next presenter will be Ms. Jordan Thomas, who is in the Lowry Lab this summer.
Good morning, everyone. My name is Jordan Thomas, and I'm a rising senior at Frostburg State University studying mathematics and biology. This summer, I had the opportunity to work in the Lowry Lab, where I studied how Wolff-Reutemann syndrome-associated genes affect gut development using Xenopus laevis. So Wolff-Reutemann syndrome is a human neurodevelopmental disorder characterized by intellectual disabilities, delayed growth, and seizures. Among the most common ways WHS is diagnosed in patients is through a set of distinct craniofacial abnormalities, commonly referred to as the Greek warrior helmet. This phenotype consists of widely spaced eyes, high arching eyebrows, and a short philtrum, which is the space between your nose and mouth. In addition, WHS patients consistently suffer from defects in many different organs and organ systems, including the heart and urogenital system. Another organ system that patients consistently suffer from defects in is the digestive tract, and this is what my project aimed to investigate. Among the different developmental issues patients consistently suffer from are malrotation, or when the gut is less coiled than it's supposed to be, dysmotility, or when the muscles of the digestive tract are uncoordinated or move with the incorrect speed, and the poor ability to absorb nutrients. These defects cause severe quality of life issues for patients, including the inability to eat orally, and patients often require a feeding tube and often have to have surgeries to correct blockages caused by malrotation. As you can see in the two images below, the gut of a human and the gut of our model organism both loop similarly, which allows us to use Xenopus as our model organism to study the malrotation phenotype. So WHS is caused by irregularities on the short arm of chromosome 4, and these irregularities can be in the form of both deletions and microduplications. The two genes that are in what is commonly referred to as the WHS critical region are WHSC1 and WHSC2, and the depletion of these two genes is both necessary and sufficient for the full presentation of the disorder. The other two genes the Lowry Lab studies are LETM1 and TAC3, and these two genes lie telomeric to the WHS critical region or on the far arm of the chromosome. Patients vary in the different combinations of these genes deleted, and all the genes have very diverse functions, which makes it very difficult to study the underlying causes and mechanisms of WHS. So in order to be able to effectively study WHS in our model organism, we need a way to knock down or reduce the expression of these WHS-related genes. So what we use in the Lowry Lab are called morpholinos. They're short sequences of nucleotides that bind to pre-mRNA and cause it to be incorrectly modified. Since the pre-mRNA is incorrectly modified, the protein product for each gene is incorrectly produced, which effectively knocks down the genes. So the Lowry Lab uses morpholinos that are specific to each of our WHS-associated genes, and they're valued at 50% knockdown rates, which allows us to effectively model the disease. So as I mentioned, we use Xenopus laevis, or the African clawed frog, as our model organism. They are both easy and inexpensive to maintain, and their genome is widely studied, which allows us to use morpholinos to knock down the genes and study the disorder. In addition, the females lay a large number of eggs, which allows for large sample sizes, and the embryos can be grown directly on the benchtop with minimal maintenance required. Particularly important for my project is that the abdomen of the Xenopus embryos is transparent, which allows for easy visualization of the gut without dissection, but the gut can also be easily dissected out for additional analysis and characterization. In addition, the looping of the gut in Xenopus laevis embryos mimics the looping of the gut in humans, so we can effectively use that to model the malrotation phenotype observed in WHS patients. So the goal of my project this summer was to investigate how the depletion of WHS-associated genes affects gut development. So since we know that patients consistently suffer from issues in gut development and they all have irregularities on the short arm of chromosome 4, we hypothesized that knocking down the genes in the WHS region would cause gut development issues. Based on a small amount of preliminary data, we expected to see the most significant defects in the WHSC1 knockdown condition. As you can see from the pictures below, compared to the control, where the gut is coiled, the gut of the WHSC1 knockdown condition is very linear, and there's also some swelling that has occurred. So to go into a little bit of background about gut development throughout the stages of development, so these are all ventral images, so the embryos are lying on their backs, and the big black spots are their eyes, and the white structure underneath is their gut. So beginning at stage 41, 1, the gut is simply rectangular in structure, and then moving to stage 42, it begins to form one loop around the top. By stage 44, it has 2 coils, and at stage 45, it's one complete coil. So since the gut is fully developed at stage 45, that's the stage that I decided to investigate the gut development issues at. So to begin an experiment using Xenopus, on day 1, the female frogs are injected with the hormone that induces them to lay eggs, and then 24 hours later, the eggs can be collected, fertilized in vitro or in a glass dish, and then they can be injected with the morpholinos to knock down our genes of interest. Once they're injected, they're left to grow directly on the benchtop until they reach the desired stage of development for analysis. So as I mentioned, the stage that I wanted to investigate at was stage 45, which takes about 4 days to reach post-fertilization. Once they reach that desired stage, the embryos are fixed in 4% paraformaldehyde and washed and stored in a buffer solution, and then they can be imaged and dissected over the subsequent days. So to get into my data, the first thing I wanted to do once I had a complete set of stage 45 embryos was do a qualitative assessment on if the embryos either had a phenotype, so their gut was miscoiled, or if they didn't have a phenotype, so they were normal. And what I noticed when I looked through all of the different conditions was that while most of the WHSC1 knockdown and TAC3 knockdown embryos were coiled normally and very similar to the control, many of the WHSC2 knockdown and LENM1 knockdown embryos had miscoiling issues. So the next thing I wanted to do was count in each of my conditions the number of embryos that had a phenotype, so they had the gut miscoiled, or they didn't have a phenotype, so they were normal. And this graph shows the percentage in each condition of those two variables. What I found was that compared to the control, the WHSC1 knockdown condition had a significant decrease in the number of embryos with gut looping issues, while the WHSC2 knockdown condition had a significant increase in the number of embryos with gut looping issues. And then we also found that LENM1 knockdown and TAC3 knockdown conditions had no significant difference compared to the control, but the LENM1 knockdown was very close to being significant with a p-value of 0.052. So the next thing I wanted to do was try to figure out if these observed gut looping issues were due to an overall shorter gut length or due to some mechanical issue in looping. So in order to do that, I had to dissect out the guts of each of my embryos and uncoil them using small insect pens so I could get images for quantification. Once I did that, I used software called ImageJ to trace the length of the gut and get measurements for the lengths. So based on the data in my previous slide, what we expected to find was that WHSC2 and LENM1 knockdown conditions would have a significant decrease in gut length because they had the most number of embryos that had gut looping issues, while WHSC1 knockdown and TAC3 knockdown conditions would have no significant difference compared to the control since they didn't have any increase in the number of embryos with gut looping issues. However, that's not exactly what we found once we normalized all of the gut lengths. So what we found was that compared to the control, the WHSC1 knockdown actually had a significant increase in overall gut length, and WHSC2 knockdown did have a significant decrease in overall gut length. We also found that TAC3 knockdown had a significant decrease in overall gut length. And lastly, we found that LENM1 knockdown had no significant difference compared to the control. And as you can see from our array of data, it's highly variable in bimodal, with half of the embryos having gut length significantly greater than the control and half of them having gut length significantly less than the control. So to summarize the effects of knocking down WHS-associated genes on gut development, WHSC1 knockdown results in a significant decrease in the number of embryos with gut looping issues and a significant increase in overall gut length. Next, WHSC2 knockdown results in a significant increase in the number of embryos with gut looping issues and a significant decrease in gut length, while LENM1 knockdown results in no significant change, although it's close to being a significant change in gut looping issues and no significant change in gut length. And lastly, TAC3 knockdown results in a significant decrease in overall gut length, but that significant decrease is not due to obvious issues in gut coiling issues. So moving forward with this project, there are a lot of things that I wish I had time to investigate further. Primarily, we want to replicate our existing experiments to get larger sample sizes and more data. So one of the issues we ran into is that the knockdowns we use are semi-lethal, especially at the later stages of development like I was investigating at. So we want to just continue replicating to try to get as large of sample sizes as possible. We're also particularly interested in the LEDM1 knockdown condition, so we want to figure out why that data we found was so variable. So that's another reason why we want to get larger sample sizes, especially for this condition, to try to figure out what's causing that. And we also want to focus on the TAC3 knockdown condition and the WHSC1 knockdown condition. So for TAC3, we saw a significant decrease in gut length, but those issues weren't due to obvious issues in gut looping. So we want to figure out if there's some other factor we haven't considered yet, or maybe it's just due to the low sample size we have. And we also need to focus on WHSC1 because it caused a significant increase in gut length and a significant decrease in embryos with gut looping issues when that was actually the opposite of our initial hypothesis. So after completing this project, our new hypothesis is that WHSC2 is the strongest candidate for a WHS-associated gene to cause the malrotation phenotype in humans with WHS because it causes both a significant increase in the number of embryos with gut looping issues and a significant decrease in overall gut length. So I'd like to thank my PI, Professor Lowry, for all of her support and encouragement, my mentor Eric Snow for all of his help, and the rest of the WHS team, Leanne and Kathy, and KG and Nick, and then the BCRU program for giving me this opportunity, and NSF for funding, and all of my supporters back home for helping me get here. Thank you.
So one thing I didn't quite follow, like how much does gut length vary on its own if you just do nothing to the frog?
From our control, you can see that the data is really clumped around the average. There's just like a few outliers, so it's normally pretty consistent.
Does that have to do with like the stage of development at which you're measuring?
Yeah, so we did make sure that we had all stage 45 because that was another issue we ran into with staging. It can be really difficult to tell what stage of development the embryos are at, but we made sure that we had all stage 45 to make sure that wasn't a factor.
Did you ever think about combining the knockdowns, doing a double knockdown?
Yeah, so since I was kind of like the first one to really go into the gut development issues, so we just wanted to do like an initial assessment of what each of the genes do individually, but in the future I think they are going to like start doing maybe combinatorial knockdowns to see what different combinations do, because that is part of the disorder in humans.